And we're live. We're back on stage with the panel discussion on AI for personalized pharmaceuticals. And to start us off, what I like to do is I like to have the individual panelists self-introduce themselves in alphabetical order, starting with their last name. So, kicking us off will be Edward. Edward, pleasure to have you again.
Thanks. I'm Edward Messick. I'm the CTO of Polycipher Health. My background is mostly in bioinformatics, but I originally studied pharmacy at the Ernest Miller School of Pharmacy, Rutgers. So I have a particular interest in pharma and drug development. And after that, I studied cell bio and neuroscience and computer science and went into genomics and then some personalized medicine at this company, Gritstone Oncology, and then worked in genetics and even in cosmetics for a little bit. And now I'm back to doing disease prediction using genomics. And I'll pass it over to Ravi.
Hello, I'm Ravi. I'm the CTO for Datum.bio. We're a connected health platform that takes biomedical knowledge bases and then makes it useful for different types of health applications. And my background is in building biomedical knowledge graphs. Um, so I basically currently look into pharmacogenomics, where I take like drug data, genomics data, um, and guideline data to be able to kind of do um, recommendations and predictions for different types of applications in pharmacogenomics. Um, uh, Happy to be here. Looking forward to a great discussion.
Thank you,
It's great
Ravi.
to meet you. I'm Dr. Shaista Hussain. Everyone calls me Dr. Shai. I'm a clinical pathologist by training, a machine learning engineer at the moment, running a company called SafeCheck.ai, where we look at the safety and security of AI systems, and from data to model to output. And it's a pleasure to be with you all in this discussion that I think will be highly energized considering our overlap.
Excellent. Well, I hope that sets the stage for the diverse perspectives that we're going to have. Maybe we can start out by giving a little bit of context for people who are not really well versed in bioinformatics and genetics and all these different related topics. So perhaps we can start with defining genetics and DNA and these things that we're born with. Edward, would you like?
Sure, I can hop in. So I guess I would start at DNA. It's kind of the blueprint of the code of our cells. DNA is composed of genes, and it holds the instructions for building specific proteins through a chain of events. Basically, that chain is DNA is transcribed to mRNA, which is then translated to a protein, and that folds into a functional structure. There's other steps in there where things can be modified, but basically those proteins throughout your body and within your cell can bind things or create functionality in the body. So one example would be like an antibody, monoclonal antibody would be specifically binding a pathogen or something from outside your body, and then ushering that to your liver to something else to destroy it, for example, or disabling its functionality. That's like one example. But anyway, it goes DNA to RNA to protein.
And I guess we've got amino acids beneath that, which build those proteins. And we've got, as you mentioned in your talk, that the Nobel Prize went for the amino acid sequencing and visualization. So I think that's a great starting point to discuss the impact that the technology has had. In that, when I first started studying medicine and pathology, we used to do chromosomes on pieces of paper and literally laid out on large tables, and we would identify the P area or Q area within a chromosome. And the rate limiting step in our knowledge was we didn't have the hardware or the software to make that process easier. And as hardware and software became more accessible, and the technology became available, we then became able to process things. But then we had the data issue, and that was one of the early challenges in big data and genomics. Was you had these like 40,000 rows and cells of Excel sheets, and Excel used to crash. I guess Ravi, you could speak a bit to that in terms of how managing large genomic data was a big challenge for a long time. And now with the systems that we have, I mean, you can take your laptop or a desk PC and process this information. There's a lot of AI softwares that have come out more recently that can do the job of 10 and 20 data scientists in an instant, and then create visuals and even tell you what are the best visual graphs. So I don't know, Ravi, if you have any points on that, on how we're marrying the latest AI data analytics software with amino acid and genomic studies. And just before you start, I'll tell you, I did an early study on sequencing amino acids using sentiment analysis and natural language processing. And we were able to do that several years ago with pretty high accuracy. So I presume by now the development of making amino acid sequence predictions and then the associated diseases with it has advanced a lot. But Ravi, what do you say on the data management?
Yeah, I think like uh, particularly if you take something like knowledge graph, that's what it's built for, where you can take like different sources of data, right? So in this case, like amino protein sequences, and like link it to different like mechanism of actions that affect like the drugs. So uh, you can not only have those data in volume but also in variety now, right? Like kind of linked across different silos, right? um, so I think yeah, I speak more from my background with knowledge graph, so being able to
What's
kind of
the GPU usage like on something like a genomic study? Maybe not genome wide, maybe a neuro panel or specific panel of genetics. What would be the energy consumption or the GPU or TPU usage to do a single analysis?
yeah, I mean it's going to be fairly demanding, right? Like because you're looking at explosion of parameters when you combine all this, like particularly with what Edward is trying to do, uh, trying to take like uh, polygenic risk scores and also across more longitudinal health data. uh, so you can really see the explosion of parameters, right? So the more parameters you have, uh, you know the more GPU power computation you're going to need with that. Um, uh, but in terms of like accuracy, what is your experience, Edward? Um, like what type of data, like you said, like adding more things, more uh, patient health data actually improves over the risk score that you're looking at outside of genomics, right? Um,
Yeah, yeah.
so
Well, I would say the way that I'm adding them in now doesn't really increase the computation too much because we pre-train the priors. But one
mhm.
day combining them would be add computational need. But I guess earlier in my career, I worked on microbiome because you're saying multi-feature models, like just like polygenics, there's thousands. Well, I was working on microbiome models and building like a K-mer index to database. And I thought at the time this was an insane compute requirement. I had a terabyte of RAM that I used to load in
Mhm.
all of the genetic sequences, and I thought that was insane. And that was like, I want to say, 10 to 12 years ago. I don't think that's even anything now. So where our compute power is just growing. I don't. I guess it is a challenge, right? They're building these data centers and stuff for LLMs. So
Mhm.
from my perspective, I've just kind of scaled up on cloud compute as big as I need, and I just kind of throw as much at it as I need. But I'm seeing with the LLMs, they're building their own data centers to do this stuff. So
That takes me to something we were talking about in your last talk in terms of the equity, though. If you talk about the fairness of accessibility to this technology, and speaking to what you're trying to do specifically, you want to collaborate with the whole world, essentially. Yeah. And you want to get even the genetics of a tribe that's been untouched, and a developed nation, and then the diasporas in between, and the genetic variations in between. And then we've become such a multicultural world, so that people are most people have some mixed ancestry,
Yep.
and more so now than before. And I guess that might complicate to some extent the genetic studies, because where if we could have taken this technology back in time, when people hadn't traveled across the
Right.
Bering Strait from Siberia to America, or whatever it might be, we could have allocated and stratified a group and said, you are at risk of this and you are at risk of this.
Yeah.
But
That's a great point. I mean, the data is siloed, so you can't really like pull genetic data together and train it together. In most cases, these databases, you have to access them on their platform. They're costly to access, so you have to use other methods to ensemble them, or just keeping them separate and pairing the genetics. But like you said, so many people are mixed, and we're using like these race labels that I would say come from the US, like white, like I'm Italian and Scottish and like four other things. You can just like, if you run ancestry.com or 23andMe, it can break down those percentages, right, pretty accurately. So I guess Europeans aren't as diverse, but like in Africa, it's extremely diverse from one country to another. They're almost completely different, almost as far from each other as they are from Europeans in distance mapping. So that's interesting.
From a machine learning point of view, that creates an issue, because your training data then would have to be very well annotated and stratified prior to running a predictive model, such that after deployment, you would want the model to be able to discern what points are worth extracting for association.
Yeah.
So I wonder in that case, how many layers of algorithms would be required to make those predictions, whether you'd use like a k-means and a clustering for one segment, and then an NLP for another segment, and then an association for another segment.
That's a good point. I think right now there's a lot of good statistical genetics done, right? And so they've built these panels that are kind of commonplace, but they can be improved. There's 1,000 genomes that everybody uses, but like, I think it should be more expanded, and then we should improve the algorithms. I think people just typically use PCA and project onto that for assigning ancestry. So it should be a little more nuanced. And I think I'm always a data guy over an algorithm guy. Like, expand how much data you have over like necessarily your methods, but both, obviously. But yeah, I don't know if that answered your question.
Yeah, totally. Ravi, what have you seen in terms of contamination? I don't want to use the word contamination, but in terms of stratifying subgroups and subgroups, because I feel that can go until infinity, right? Like if you, based on the mixed populations, or if you're in Edward's case, combining phenotypic features like lifestyle and diet, etc., and nutrition. To what end do we stratify until we get the most predictive markers to use in training, and then be able to say, okay, now we've got a good personalized medicine picture, and now we can start doing pharmacogenomics? Because if you mentioned, Ravi, you're in pharmacogenomics.
I thought of it.
Yeah. Oh, let's hear.
I'm sorry. I'll go back to Ravi because he's like the guy who combines this data. But the way that we do it from a clinical perspective is based on deviations from the mean. So 2 standard deviations beyond the mean, or a certain percentile where their risk multiplier is great enough, right? That's how we're doing it right now. So it's
just to pick your brain before we pick Ravi's brain, and maybe Ravi, you can jump in on this. if you're basing it on the standard deviations of the mean, and you've set a mean based on an aggregated data corp, right? When you're processing your data or pre-processing it, you're definitely going to have missing values, imputed values, null values. And then if you're going back to the mean, then you're almost biasing that mean so that you make the data fit, and it tends to exclude outliers. But it's specifically in the case of health, in health AI, outliers are so important because it's from a subpopulation. So your data sample, one outlier might actually represent a whole nation of people that also have that outlier. So how do we converge that, Ravi?
Yeah, I think I guess from the machine learning perspective, it's fairly simple in the sense like you're basically kind of uh, doing a feature selection and then kind of coming up with like the number of like stratification you want to do, right? Um, but the question more is like in terms of what Edward pointed out, like is it something that can be clinically uh, used, right? And and I think we have to meet in the middle a little bit, like uh, uh, instead of going like top down approach, maybe also bottom up, where you're actually using the domain knowledge for that. For like for example, in pharmacogenomics, like when you really look at phenotypes. They're heavily dependent on the functions of the alleles or whatever, right? Kind of, is it like, decrease function, increase, and then they have certain way to kind of do that. So we could take advantage of those specific domain knowledge to kind of uh, incorporate into your algorithm to be able to do better. Instead of just like relying specifically on the algorithm itself to come up with the stratification. Um, I think also a bottom up approach of actually starting from domain expertise can lead to better results. That is actually more clinically relevant. Um, yeah,
But where are
just
we
based on
getting
my
these kind of data sets? Where are we getting these comprehensive data sets? So, I mean, the few I did a couple diagnostic models on acute lymphoblastic leukemia, for example, or breast cancer, hepatocellular carcinoma, and Edward, you might be able to relate to this, and we'll pick on you, Ravi, for resolving it for us, I guess. But getting those data sets was the longest journey of our ML ops. So we'd said, for example, we can do this in 8 weeks, based on the data and the modeling and experimentation, baseline, etc. And it took us 8 weeks just to get permission from a single university somewhere in the middle of nowhere to give us the slides or to give us the information. And then we ended up with bias because our population was so small. So where are we getting these comprehensive data sets to be able to do all of these benevolent things that we say we want to be able to do? It's tough. Who has them?
Well, just depends what you're looking for. I'm sure that's what Ravi does, right? He pulls together all these ontologies and databases. There's a lot of them, but
it's challenging
it's
to
hard.
get all
Yeah,
of them. Yeah.
it really is. That's like the biggest problem is that even for these government run ones, the restrictions and the access to just a research institution to keep away from commercial interest. Then you have these industry companies that built their own, and then you have to pay, right? But there's certain ones that kind of, like I like KEG, right, for pathways, but they pulled it away and now it's back out there. It's like it's tough, but I'm sure Ravi has good comments on that because that's what you do, right? You're pulling together and building knowledge graphs, and right?
Yeah. And one challenge is also like, even if the data is available, it's not necessarily in a very standardized format. Like,
Yeah.
it's very difficult to put it in a format where you can actually link it to your data or to like other knowledge bases. Um, because like I think when they're published, like they don't necessarily think of uh, it being used beyond their immediate scope of use, right?
Yeah.
Um, so they're not isn't necessarily bothered with like
Uh,
making them fair.
And
Sorry,
then
yeah,
we've got
please.
regulations coming up from everyone and their mama around the world is popping in some new rule regulation that it's
Hmm.
suffocating the not only machine learning creative potential, but the access to the data potential is it's being suffocated. So I think
yeah,
this data acquisition and data use is, I guess, the biggest bottleneck at the moment. Now, some of these compliances, like the EU AI Act, marks healthcare AI as a high risk category, and immediately that means back off, or you
yeah.
lose it.
Yeah, I mean, I'm facing some challenges with grants for like polygenics, even, which is just a basic linear model right now, and it's like we just want monogenic still. So even academia or scientific grants, which should be advancing things, are kind of living in the past. They're kind of dinosaurs when it comes to AI. But I think it's really interesting what you're asking, like, or what you mentioned, Ravi, that they're not standardized, right? So, like, we can use LLMs maybe to standardize these things and to bridge the gaps in different ontologies. I'm kind of facing that with electronic healthcare records. They're different codifications of disease. There's different, even the doctor writes it differently, transcribing their handwriting. I mean, I was a pharmacist for a little bit. That's impossible. I mean, you guys
Hmm.
must learn in med school to write messy on purpose to mess with people. But there's so many things where I think AI comes in
There's
with
a
the
good reason
standard.
for it. They give you like 10 minutes to answer 3 or 4 chapters worth of information. So you get
Yeah, I
used to
know.
being in a rush.
I know. I'm just joshing. But I think LLMs can like bridge a lot of those gaps of standardizing things, pulling out information from records that aren't in text format, for example. And a lot of companies are doing that with EHRs. I just kind of dove into that field, so I'm bringing it up. But with the ontology stuff, it's super interesting. Syncing databases that may be calling one gene a different um, gene in another database. So, is that what you're doing, Ravi?
Yeah, I was going to ask.
Uh, Pretty much, yes. Like, and knowledge graph in particular are very good tool for that, like, because it helps to layer in your data, like starting from ontology to like, your knowledge base to like, like if you start from disease to genes to like, it's very easy to kind of uh, jump from one domain to the other in a single database, which makes it like it almost has a simplifying effect on the types of application you build on um, top of it. So
Yeah,
including also for language models.
yeah,
One
structuring it properly.
Mhm.
one of the earliest
Yeah.
adopters of knowledge graphs and semantic technology was the life science and pharmacology space. Ravi,
Mhm.
possibly you could talk a little bit about how you're integrating all these different fragmented views of the same kind of ideas. You have the governance of a particular substance or disease. You have the treatment of the disease in a healthcare setting or a clinical setting, and then you have the biological study of that protein or substance or how you're diagnosing what that is. So maybe you could talk a little bit about that entity disambiguation.
Sure. Yeah, I think like Joaquin pointed out, like healthcare is, I guess, one of the pioneers of I think even before we had officially the term ontology, um, like the Human Genome Project was already working on something similar that turned out to be the gene ontology, right? Um, and like uh, healthcare kind of has an abundance of all these knowledge bases, um, and they were kind of the pioneer of kind of figuring out how you link, uh, at least initially, how you normalize different concepts into like one, like if you're aware of like UMLS, Unified Medical Language System, it's been a long-term effort of like NIH, where they take like concepts from different vocabulary and try to normalize into like one like a vocabulary, um, so that like if you're trying to do name entity recognition in like let's say a clinical note, um, like even if it says MI or myocardial infarction or whatever, it can be resolved to the same concept if it's related, right? Um, and recently, like with popularity of knowledge graph or graph structure and language models, um, like people are building graphs out of like documents, but these are like more text graph where they're basically kind of linking text to one another, right? Um, but I think more powerful is what Joaquin just talked about, like being able to resolve those entities into an underlying ontology, which links it further to like other related subdomain. So your drug to your gene, so you can kind of see directly the interactions between drug gene, and you can then layer on top like more very specific domain knowledge, like what comes from from GKB for in case of like pharmacogenomics. Uh, so uh, and graph databases, particularly semantic triple stores, are pretty well suited for that. I know you can do those in relational databases as well, and but like graph databases like are pretty much built for that, and they work pretty well for that purpose. And almost they simplify it the things that you're talking about, kind of challenges with accessing data, linking them together. Knowledge graphs actually make them very simple, so it has like very powerful implications for AI downstream.
It's very interesting that you talk about the format of these databases and then like the knowledge they're linking to, because on the base level, a lot of these link to actual lab results, actual measured results, like with protein database. They have like, they run cryo and they actually resolve the structure of proteins, which were used to then train and validate like all these models, like Google's DeepMind and Baker's stuff. And then that goes all the way up to the level where you're structuring your actual database for text and LLMs. So it's pretty interesting how there's these different layers of that. And it's funny that I think a lot of these databases, when it comes to bioinformatics databases that aren't necessarily ontologies, it's data, genomics or whatever, the way they're stored isn't necessarily in a good way to train data for models. Like they're stored just in a disparate way, or they're stored in one giant flat file. It's like crazy kind of. So that's a big overhaul that I think you've highlighted.
Yeah, but then you've got these new AI data science models that you can additionally add to that layer. So you've got the LLMs, you've got the knowledge base, and then you've got these AI data analytics that are, in my limited experience with them, they're super efficient in terms of aggregating the data and then stratifying it and finding things that you would otherwise as a human team miss, which is a nice relief. So I think there's like multiple layers of models that can be used to optimize the entire process. In the end, it looks like we're heading towards multi-agent systems to achieve it. So, and I know the whole agentic workflow can be kind of controversial, but in this case, it seems like the logical solution to use agents with different features. But then that comes with a risk, and of course, I'm going to bring up the risk of the agent. In that, I don't know if any of you have worked with agents or agentic models. You don't know how they're doing what they're doing. So you basically create a persona and you say, okay, you handle the data science, you handle the data analytics, you'll handle the knowledge graphs. And what are they doing and how are they doing it? You don't know. You really have no insight. So that's another major risk. What could go wrong is infinite.
Do you think like layering them, you kind of have another agent that checks on it, is a QC step? Is that the
Yeah,
solution?
definitely. And you can have a QA, you can put in a quality controller, a quality assurance agent, but even then, you don't know how he's doing what he's doing because they're speaking to each other.
Right.
Like getting you're in school and then the brats go to the corner and say, okay, we'll get the job done. They get the job done, but did they steal it? Did they cheat? Did they cut corners? You would never know. So I think that's an upcoming risk to consider in healthcare AI, given the challenges that Ravi you highlighted and Edward or Ed, I decided to call you Ed, and that you brought up. There are these challenges that are slowing us down, and then at the same time we have technologies and new models that can handle it, but then they come with their own risks. At some point, we may become dependent to achieve these goals that we've been outlining on these agentic workflow systems. And then when we do, we really lose control. And that's when I think the matrix happens, or like some next dystopic step, because you now depend on them and rely on their predictions and their model outputs, not knowing how they achieved it. At any point, if that becomes contaminated or malicious, whether human inserted or maybe there's a bad robot, so to speak.
Well, how do you know we're not already in the matrix?
We are. We might be.
Based on simulation theory, if it can exist, the chances that we're on the base layer infinitely small right now.
Yeah.
So
At least it would
let's
explain
embrace
deja
it.
coincidences.
Hold on, I'm gonna go try to fly out of my window now.
So
Life.
we sort of addressed the foundation of this conversation around genetics and the composition of how people are built with proteins and how they engage, how we manage the information of all these underlying components. Let's talk a little bit about a little bit more practical sense of we're living here. You know, we are in different places. We eat different food. We have different diets, and we are exposed to different environmental factors. Some of those things agree with us. Some of those things cause dis-ease. So I don't know if we want to tackle this. Shai, maybe you could start us off with
I'll
your.
start us off and then I'll pass the mic to Edward. Dis-ease, as you mentioned it, yeah, it's a state of non-homeostasis. It's when we're not in our optimal metabolic functioning processes. And then when we look at nutrition studies, one of the areas that we failed at previously, or not failed, we were less than optimal, is we were doing studies and finding associations so as to say, okay, low magnesium, you might feel very tired. So then people get magnesium rich supplements. And then they found that the extremely concentrated magnesium can have an impact on muscle contraction. So where there's not a fully comprehensive understanding of which components within a diet play to help or hurt, and I guess that's the basis of medicine, is it can heal or it can harm. But where there's a lack of understanding in terms of concentrations and dosages and impacts, you actually create a new negative cycle where you may have just been better not introducing these new fads and trends altogether. And that's another role. I'm just going to throw in AI in here and machine learning, because that is a role that we would otherwise be blindsided to. And then as these data aggregates grow larger and larger, like with what Ravi's doing and what Edward brought up, we might be able to pick up on data points that otherwise we would have completely overlooked, especially when it comes to disease diagnostics. So we have these like correlation maps, these heat maps, where we take from our data set and we say there's all these data points in this CSV table that we have. And we're very keen to look at disease positive, disease negative, and then we look at gender, for example, or age, and we look at the very most obvious systems to be able to get a publication. I mean, that's typically the goal. And then you say in your publication, we found that men aged 15 to 30 are at higher risk of some particular like nail fungal infection, or it could be anything, whatever it might be, where we as humans would have overlooked, as we mentioned earlier, the outlier points. Machine learning would pick up and say there's an association with male pattern baldness that is contributory to this, or whatever it might be. So I think that the concept of understanding what causes dis-ease or disease in general the light has shined on us, and we have this opportunity henceforth to make more comprehensive predictions. All that said, and my final point before I hand the mic to Edward and to Ravi, if we don't correct the imbalances in the distribution of data, so if data is not fairly distributed, bell-shaped, with agreed upon medians and standard deviations from these data sets, we're just going to perpetuate the bias, and in which case our technology is going to hurt us way more than it could ever help us.
Wow, there's a lot to touch on there. So I'll try to I was writing some things down. So I do think there's an awesome place for LLMs and for AI to come in and do these meta studies, to even find studies that may just be sitting out there. A big problem with nutrition studies is they weren't done over long enough periods of time. They weren't controlled well enough. They didn't factor in lifestyle. Like a lot of them do that, right? But most studies are junk. But there are these multi-decade studies that give us some good trends and some good information. What most of them, if not all of them, I don't know if I've ever seen one that includes genetics. So nutrition is highly genetic. There's a microbiome component, which is like really hard to even tease out in the study these days. I did a blog for microbiome for 6 years, and I've read so many studies on it, and it's massive to nutrition. It shifts due to it, but it also causes your reaction to it. So everybody knows somebody who can chug Gatorade or Kool-Aid all day and stay skinny. And then, so it's not only your lifestyle, there is a genetic component. It's not just calories in, calories out, in my opinion. There's age, there's so many other factors. Do they control for all of these things? Not necessarily, but there's some evidence. I look at blue zones for nutrition as well, where people live longer. So I think what we're finding now is like mostly plant-based. I like to eat a lot of protein, so I'm not going to stop eating meat personally, but, and then maybe eating less, fasting is proven. I mean, all this stuff, you look at C. elegans, model organism, or any model organisms, they live longer when you restrict calories. So those are some trends that I'm seeing, and there's genetic basis and there's pathways in our body that explain like why that would work. Can we leverage that in pharma is a super interesting question that a lot of companies are starting to address now. The longevity and aging field's getting tons of funding since the pandemic. And before that, there's some pioneers of that to address pathways of aging, which should address a lot of other diseases of aging. And now these GLP-1 inhibitors, Ozempic, all this stuff is having huge impacts on people's health. So like certain pathways you can really leverage, but I think like the most powerful medicine is nutrition and is your lifestyle, because you're not triggering these like negative pathways. And that made me remember that a lot of these studies you're comparing to like a Western diet as your baseline, which is like junk, right? And it's not standardized. There is no mean to normalize to. Like one guy's eating McDonald's every day in the Western diet, another guy's eating, steak and potato, like whatever. I mean, so it's to define a diet, even if you look at like the ketogenic diet, which has been used therapeutically, the definitions of it across studies vary so much. It's insane. Anyway, that's just something that I've studied personally because I just love to read science, but I'm not an expert in nutrition. So
yeah, you are. Yeah, you are.
Well, maybe, but I think just the genetic element is super interesting to add into those studies. Will they ever get funding to do that with microbiome in an accurate way for 30 years straight? Like probably not. So can we get that data?
You mentioned it's dynamic. I mean, even the diet consumption is dynamic, and then going back to the data points, that makes the data analysis dynamic. So one of the things about in clinical pathology using ML, you take these screenshots, the snapshots in time that are static. And so you say the average person from this area has red blood cell or white blood cell parameters of this to this. That was on that day. And literally an hour later, those parameters would have changed. And then 2 days later. And then what we are doing is we're building AI models based on static screenshots. So I would
Yeah.
love if we had dynamic longitudinal data sets that could
Hmm.
feed into a training system that's dynamic rather than a static. I don't know, Ravi, am I dreaming to think that
Mhm.
we could really have
I mean,
dynamic training sets?
no, I think that's probably where we are heading, right? I think that's what Edward is working on towards as well, like in terms of taking, um, you know, so many parameters and make it very specific to a patient. Uh, because right now, like we are following guidelines, which is kind of compiled through a limited set of evidence over it takes like a couple of years to compile that evidence. Um, but if once like the AI becomes very good at taking like more parameters than these guidelines are considering right now, maybe that's what the future is going to be. And I think that's what Edward is working towards as well, being able to just take you know a specific data of a patient and
Hmm.
be able to predict very accurately. So
Yeah, like
so yeah, I think maybe we're not. Yeah.
reintegrating the data in like a healthcare learning system. I didn't really speak on it in my talk enough, but I think touching on what
Mhm.
you're saying, longitudinal studies, like there's a big push for epigenetics, but it's like you said, when you do just sampling or a different time of the day, or the guy walks by and smells a guy smoking outside, then he goes in to do like it completely could change
Mhm.
things. So it's really hard to normalize that. Maybe that's where AI comes in, and you can kind of
Mhm.
normalize these longitudinal data sets with all these other factors better. Um, but
Sensors too,
it's
like the IoT sensors are going to play a role, I think, in that regard. We didn't touch on that, but
oh
like
yeah,
you just said, the thing about the guys that walked by someone who's smoking, or if you were to take a painkiller, I guess you guys have Tylenol there, we have Panadol here, but I mean, if you were to take a simple painkiller for your headache, and you take a blood test within 48 hours, your liver enzymes will be very abnormal from that single tablet that you took. And then, like
right,
in current medical practice, one of the laughable moments is when you get, well, as a pathologist, I would have physicians say, "Oh my God, this young kid has really abnormal liver enzymes." And then by the time they come to the lab, and I'd ask, "Did you take any painkillers or anything for a headache in the last 2 days?" And be like, "Oh yeah, 2 days ago I did." I'm like, "Well, there you go. I mean, we don't need to explore this any further." But if you have these sensors, I guess we're becoming cyborgy for a good reason. But like if you have
yeah,
these sensors sort of embedded, and then it's detecting that you've just taken a tablet, and then now your enzymes are abnormal, and there was sort of a lifetime feed for that, we'd have a
right.
much clearer picture for association and causation of things.
Yeah, there's too much human error and missing data on things.
Mhm.
I also think it's like interesting that just like natural changes, like circadian rhythm,
Yeah.
throughout the day when you're sampling. So that's why I like to focus on genetics because it doesn't change really. I mean, epigenetics does expression things downstream, but then you bridge that gap with like those snapshots of that over time. Um,
You bring up the circadian, which is a good point, and I do a lot of research on circadian studies and sort of neuropsychiatry. And one of the things I found just in my own experience is, again with the disparity in data, Ravi, again with the disparity in the data, circadian rhythms innately are hormonally based, so such that you get sort of sunlight through this receptors in your retina, which speak to the suprachiasmatic nucleus, which speak to the pineal gland, and then they communicate back and forth to the hypothalamus, and then they say, yes, you can sleep, no, you can't sleep. So it's this clockwork mechanism that happens, but the communication happens through hormones, which are inherently different between men and women, and then children, adolescents, adults, and seniors. And so there's already so many shifting data points that can influence the predictions. And lo and behold, the current recommendations we have for example, a lot of sleep disorders are pop a melatonin, or pop this one thing, and that's
right.
based on a very specific subpopulation within a data set where a study was done. And I'm really hoping AI studies can can figure that out where we've been missing on it.
Yeah, I agree. And I think there's a lot, maybe we can shift to pharma, right? Because there's companies that I've been a fan of for a long time. There's this company in silico medicine that has done really cool stuff in that way, in digging through the clinical trials, finding drug targets, new drug targets for existing drugs, and generating new molecules. And a lot of it comes from finding those gaps that you're talking about. So I think that's like we have this deluge of, we have so much data, it's not perfectly annotated, there's missing gaps in it. How does AI come in and fill in those gaps? You know?
Ravi, how are you managing that? Like, how do you manage those data sets? And I know that when there's imputed values or missing values, I'm not a fan of people just taking the average of the existing data, but how do you manage that?
Um, yeah, I think one way is definitely like uh, simulating uh, based on like known uh, you know uh, uh, parameters, right? You can uh, uh, simulate some data, but that's probably the closest we could come if it's missing. Um, or like you can substitute in like the closest proxy, like yeah, I see.
How?
Cecil. Oh, yeah.
Doesn't that cause a
Um,
bias though?
yeah. I mean, yeah. That's bit like I mean, um, it's good enough to get idea about like what the data should be, so that uh, like you don't really want to use that, but just to get an idea of like uh, what should be filled in the gap there, right? Because like even if you take two clinical trials, they would be like looking at two different things, even if it's for the same disease, for example, right? Um, so um, for your third clinical trial, you could take a look at those two priors to see like what type of data they really collected, what can predict from it, to kind of get a sense of like what how we should design this clinical trial, for example, right? Um, so it obviously is not going to be applicable for all problems, like especially if you are really trying to build like a predictive model that needs to be accurate. That's probably not how you'd want to approach the solution, right? Unless you know like the impute method you're doing is very good, right? Is very applicable for that particular use case. Uh,
Ravi,
Yeah.
but
I think I can
yeah,
touch upon, like, because it made me think of, we're dealing with a missing data problem. Can you use AI to fill in data to then train other models? It's going to increase bias. But
You mean
in
synthetic, or we're finding the median and substituting?
probably both, right? I think. But
mhm,
what the example I thought of was, of course, coming from polygenics. It's where people, or imputation rather, people have done the work, the statistical work, to add another layer of information about these genes for imputation. And what they use is linkage disequilibrium of those genes to other genes. So then when you're imputing these other genes that are missing, you have other data that backed that disequilibrium calculation. So and linkage patterns that are found in populations. So maybe you can do some statistical studies from other data sources and bring them in to then project those
mhm,
better, like out of cohort type of training, you know?
Ravi,
mhm,
maybe you
mhm.
could talk about some of the work that you're doing at Datum and explain how you're integrating all these different sources of data to create the backbone for further work.
Mhm. Yeah, I think we're not doing that much predictive models as such, but um, we're basically at this time like we're basically taking like uh, uh, to figure out like how we could take all these guidelines in healthcare. So one of them is like guidelines in pharmacogenomics, and then use them to actually make sure the models adhere to those guidelines, right? Because like um, if you're familiar with current approaches of kind of putting guardrail on language models, it's either like very prompt based, or like very much like document kind of retrieval search kind of based, right? um, so what we are working on is more like can we take these guidelines that can help to extract some sort of reasoning that will uh, help the language model to kind of stick to that guideline. So earlier when Saisa was talking about kind of agents, different agents being able to do their particular job well, um, I think that's where like the guideline adherence and the guardrails come in. So you can basically, we have a framework called knowledge augmented graph reasoner, which basically um, takes his guidelines, builds a reasoning framework that can be benchmarked using test data sets, and also um, it can be augmented with broader knowledge graph. So we also have like a broader knowledge graph which brings in knowledge from like different domains and subdomains in precision medicine and healthcare, which provides like the context the reasoner needs sometimes. So like for example, the earlier question about um, like phenotyping, like genomic profiles, for example, right? So that could be in a knowledge base, like, okay, you have ran some predictive model, you have come up with like, okay, like these are the ways to kind of phenotype a patient. You can actually then put it into a knowledge graph that can be used by a reasoner to then use it as a context to kind of look at the input data, like it can, language model can ingest all the input data for a patient and use these contexts to then reason and say like, okay, this is of this phenotype, right? So that's something we're trying to do at Datum, and we have started with pharmacogenomics. um, we are trying to kind of expand beyond that into more like precision medicine, where even if like you don't necessarily have genetic test results, you can still use like other patient data to be able to kind of give very specific, like drug related guidelines, uh,
Are you
so to
generating
speak.
new pharmaceuticals from that data? Is that drug generation a part of that process? Because seemingly that would be a logical application.
um, not at this point. We are mainly just utilizing what is there, right? So kind of um, like one of the things we are doing is like there's a lot of biomedical evidence out there, um, but it's not getting to the bedside. It's not like, it's very difficult to integrate these into the application. um, so being able to kind of put them together in a more meaningful way.
And seemingly
So
that would be
we're
a
not
great springboard for both within for clinical trials and otherwise, in terms of using that brilliant data that you're sourcing towards generating personalized pharmacotherapy for people. I don't know,
Mhm.
Edward, maybe you have something on that, but I see that as being, the personalized pill, the ed pill, the Ravi pill, the Shai pill, based on our genetics. We could generate pharmacotherapy.
Yeah, absolutely. I mean, I want to use polygenics for clinical trial stratification. This company, GlaxoSmithKline, started to do that, and that's just downstream in the actual like selection of patients based on their genetic risk. They do that with monogenics, right? You're targeting a certain disease with a known causative gene, but it's more complex than that. So that's just starting with the polygenic perspective. Um, for predicting actual, drugs, that's also starting. I think in silico medicine's the leader I see in that field. They took all these databases of drugs and they've predicted new molecules that are actually in trials. You should follow them. It's pretty awesome stuff. I think David Baker and people doing protein prediction are gonna like be the future of that, because I think basic molecules are kind of the past. Yes, they have their place, but it's like things like proteins and designer proteins that are really the future. There's even like nano cages and things for drug delivery, which are fascinating. mRNA vaccines are like a huge leap forward where they are using bioinformatics to predict the structure, right? They used it for COVID and with great results. And so I kind of did that in cancer at Gritstone Oncology. Yeah. And what they did was basically sequence the patient's genes, look at their T cell response downstream, but sequencing them and then predicting based on their HLA typing and their known cancer that they have a vaccine cassette. So they were giving them a vaccine to trigger their own immune system to attack.
Dude, this is what we should be doing. This is the gold of what we should be doing.
It's insane. Brilliant. So I only worked there for a year, but it was insane. And they also have so there's a couple companies doing that in cancer, but it's a super complex disease state. So I think there's a lot of room for other diseases for that type of personalized medicine.
Yeah, I mean, there's so many diseases that we've been stuck at from, like, you take a look at...
You're just throwing it at a population.
Exactly. Like, case in point, in cancer, breast cancer has been pretty stagnant in terms of the chemo cocktails that people get. And it's very much a trial and error. They throw it at it, and if it works, great. If not, we'll try another cocktail. And it's a lottery, and because of everything you just said, the genetics and the epigenetics, but we've been stagnant. And then outside of cancers, there's the same thing with like neurodegenerative diseases, Parkinson's. We've been stuck for so long.
MS, it's their body attacking. And a lot, AI actually had a role in, like, it's not proven yet, but that a lot of those diseases have a viral origin. You have a certain number of infections, and then it causes MS, and your body's attacking itself because it's having an immune response against some shared protein from this virus and your own like nervous system. So yeah, I think there's so much room in other diseases, but the reason is because regulatory, like you have to go through all these standard lines of therapy. Then you're already in this like crazy cancer state where it's ethical to do a trial with that. But I think that's the future, is real personalized medicine based on all these factors, where it's generating almost like a unique drug for you, rather than screening one molecule against a massive population. That does work too, but yeah, I think that's the future. And we talked a little bit before this about this doctor's neurosurgeon, Gooby, on YouTube. I'll send it to you, who left neurosurgery because he realized that the body's ability to heal itself was actually what made the difference, not his surgeries necessarily. So it was like how they ate, their salt intake, if they low stress. And I've seen that in myself with things that I've had where it's like my lifestyle changes were more impactful than maybe other things that were being suggested by medicine. Of course, those things work more, and doctors are gonna, there's more strong evidence, real clinical trials that prove that. That's why. But with this nutrition and lifestyle stuff, it's triggering your pathways in your body to heal. That could be
another point, though. That's brilliant in terms of what you said about the nutrition, but it triggers in me the fact that a lot of our modern food is chemically processed, and so like, even our corn is CRISPRed, our potatoes are CRISPRed, and then you look into the soil or the pesticides, and then there's
nitrates.
Whole discussion in terms of, because when you were speaking, I wanted to say that maybe the old way was the right way. Back in the day, people used to have a farm in their yard, and they used to make their own food.
I did.
And not so.
Not a farm.
Not that old, but like in those days. And there seems to be a logic and a brilliance to the way our forefathers lived, our grandparents and great grandparents. But then now, how much could we live like that? One, with the fast pace of life that we live in modern civilization, and two, what what kind of chemicals are we taking in from the farm, from the farm?
We might seem like hippies right now with talking about all this stuff, but it's true that there's some wisdom in the old ways. There's this thing called permaculture. I went down a huge YouTube I went down a YouTube rabbit hole, and it's basically planting in your backyard like a food forest in the patterns of a forest, and it leads to greater yield. And there's people that in my area have 0.1 acres, and they're able to feed themselves for like half the year just off the so that's going to be healthier. Like farmers markets are going to be healthier, but it's just like leveraging. It's putting your body in a position to heal. Like our body's amazing at healing. Cell therapies and these like precision therapies are the future because they're leveraging those same pathways that you can do like with fasting or, a lot of this stuff isn't completely proven, right? But there's good evidence to show in certain diseases. Anyway, sorry.
I want to jump on that, and we're at time for the panel discussion. We could easily fill up another hour if we really wanted to. But that hippie thing, that's BS. You go to isolated tribes and you look at the architecture, you look at the way that they construct their homes. People are like, oh, they're mud homes, big deal. They're breathable, they grow, they're modular, fast to construct, everything sourced locally, no carbon emissions. They last like hundreds of years, it's like insane. And the permaculture, people were talking about people being savage and living in the woods. Bro, the whole surrounding area was their supermarket. Why would you want to cut things, harvest them, and then put them in a package when you can just rip it out of a tree that you grew and architected how it's going to feed the local population?
We need to change how we do farming was like why I thought of that, because you said they're genetically modified so that they can just grow off nitrates and that they're always the same for mass production. But actually, I think in India they started to do some permaculture stuff, and they restored these like rivers that were completely dried out by like doing it, and they have more yield of every single crop. So that's just a totally different topic, but it all comes together in that it's what is triggering disease in our body. And maybe we can leverage those pathways in pharma too, directly.
That's the root of pharma anyways. The root of any tablet that you take is it's coming from some plant or from something organic, and then it was mimicked in a synthetic lab.
Yeah,
it makes sense. Our body is evolved in symbiosis with nature and with these foods. Right. So it's all
back to the basics.
We've jumped around a lot. This was a great combo. We've went from data like structures to like hippie permaculture.
And we settled on all of us are gonna get a joint farm and grow our own stuff. Ravi, looks like we're coming to you.
We're coming to
the
Nepal.
We are
up in the mountains,
fertile soil here, so
away from like the rising oceans. We can go have a homestead up there one day and hide out.
We'll be in touch, Ravi.
So with that, it's been a pleasure being able to speak with you all and have this panel discussion. We're going to have to cut it here, but that doesn't mean this is the end. If anything, this is an invitation for a part 2. So take a break, go network with people, connect with people, and we'll be back in about 45 minutes or so.
Thank you, Edward, Ravi, and Joaquin. Brilliant.
Thanks, guys.
Thank you all.
Thank you.